Hello. I'm just setting up a couple of things before the live stream can start. Okay. Mhm. Okay. Letting people join. I'll get around here. Okay, one more test. All right. Welcome everyone to this live streamed MCAT content lesson about the amino acids. So today's lesson is going to cover a topic in biochemistry that is the most commonly tested biochemistry topic on the medical college admissions test, aka the MCAT, that students need to take in order to apply to medical school in the United States. So my name is Aiden. I am an MCAT tutor. I tutor online and in person in New York City. And today's lesson is about amino acids, which are the building blocks of proteins and protein. So I am going to conduct today's lesson in the format of a document where I have an outline of the content about amino acids that I will talk about today. And as I'm going through the topic, I will explain what I'm writing in the document in real time, and I will write in the document what I'm explaining in the video. And once this live stream is over, I will post the link to that document in the description so that anyone can view the document as they watch the video. All right, so I'm now going to share my screen. I forget to do this. Okay. All right. So today's topic, amino acids. Before I start talking about amino acids, I have both reputable sources that I'm linking to and calling out before this live stream starts that I am basing this information off of. I am not an authority on any of today's foundational science topics. These have all been covered to great length elsewhere, and if anything, at this point, are outdated. So these are the links to the different more reputable sources. But before we get into that, I have a YouTube playlist that I've linked to in this video of more well-known individuals on YouTube who have covered this topic in much greater detail. And some of those videos are tailored to the MCAT, some aren't, but I believe that they all help build a stronger understanding of the topic of amino acids, which is very important on this test. So just to briefly go through these sources, Biochemistry Concepts and Connections was the biochemistry textbook that I used when I was taking biochemistry at Brooklyn College. It is Matthews, Appling, and Anthony Cahill, specifically chapter 5, which is Introduction to Proteins, the primary level of protein structure. I also linked to this Rice University Creative Commons license chemistry textbook that I use some foundational chemistry concepts from, and also their anatomy and physiology textbook, which covers some of the essential information about amino acids. Then I have one of the most important sources for MCAT preparation, which is the writers of the MCAT exam themselves, the Association of American Medical Colleges. and they published a What's on the MCAT exam PDF, which is still live on their site, which is a content outline of what the exam covers. And then finally, I have a popular review resource that is published on Reddit by a user named Miles Down, which is a 90-page review sheet that condenses all of the information on the MCAT into a very easily digestible, sometimes inaccurate, but mostly very useful review sheet. So now that I've gotten through the sources, these are the topics in the topic of amino acids that I am going to be discussing today. I want to read through this lesson plan first to give an idea of all that will be covered, and then I'm going to go one by one, adding my notes as I go along under each of these headings. And I'll also be showing figures as I'm adding those notes. Okay. Some of them will be from the sources above, some of them will be from other sources that I've gathered in looking into this topic. The first is going to be definition of the amino acid. The second is going to be the alpha carbon on the amino acid. 3 is the NH3 plus and COO minus groups. 4 is the R groups. 5 is the acid base properties. 6 is special properties, and this is all of amino acids, so the acid base of amino acids, the special properties of amino acids. 8 is the names and abbreviation of the specific amino acids that you have to memorize for the exam, which is 20 of them. This video isn't intended as your memorization resource, but it is important to at least state all of them before we're done. And then my last point that I want to cover in the live stream today is an AAMC question. You'll find a question directly asking about amino acids on practically every exam for practice that the Association of American Medical Colleges publishes. So every one of those exams is going to have an amino acid question. So I chose one that I liked and that I will discuss at the end of this video. So number 1, the definition of an amino acid. The amino acid is the fundamental component of the protein. And in the human body, proteins not only make up a large part of the structure of what makes our body, but it also is heavily involved in the function of our body. So both what it is and also how it does all of the things that it does. So the proteins that make up our body, you have the structural ones, functional ones, and then when it comes to amino acids, as the building blocks of those proteins, amino acids can serve, well, that function, the building blocks of proteins, and they can also serve as intermediates in the processes that our body goes through to make energy and to convert energy molecules and other important molecules from one kind of molecule to another kind of molecule. So the amino acid can be a sort of intermediary in many metabolic processes. That's sort of part of the importance, which I think goes along with the definition. So I'm going to try to make this a little bit smaller. Okay, so the importance, protein building blocks, aka monomers. So the mono is one part of the protein, and then the protein itself is a polymer, so multiple parts. The amino acid is the monomer of the protein, the monomer constituent of the protein, and then the polymer is the protein itself. So the protein building blocks and metabolic intermediates. And I mentioned these 2 points as their importance because these are the 2 ways in which amino acids are going to show up on the MCAT exam. They're going to show up as parts of proteins, which do all kinds of things in the body, including making up many of the structures of the body. And they are also going to be tested as sort of middlemen when the body is going through its metabolic processes. Okay. But then what is the amino acid? And the amino acid is a molecule, but how do we define amino acids as molecules? What makes them different from every other kind of molecule, specifically when we're talking about biology? So it's difficult to put it into words. It's much easier to simply show a picture of it. So this is Aiden's drawing of an amino acid, my drawing of an amino acid. I will describe what's going on in this picture. It helps to have a background in organic chemistry, but I'll explain in words also what's going on here. So this is a drawing of a molecule. It has 1, 2, 3, 4 different kinds of atoms. At least we can assume so if the R group is just a carbon-hydrogen chain. So you have hydrogen atoms, the first element on the periodic table. You have a nitrogen atom. You have 1, 2 carbon atoms, which are implied by the angles in this line angle drawing. And you also have 2 oxygen atoms. So hydrogen, nitrogen, carbon, oxygen. This is an organic molecule because it is made up of those atoms. And it turns out that among organic molecules, when you have a molecule that has this specific nitrogen hydrogen configuration, which is called an amino group, and this specific carbon oxygen configuration, which is called a carboxyl group, it is an amino acid. So to expand on that a little bit and connect it to why this is the definition of an amino acid, this part of the molecule is going to act as an acid. And this part of the molecule is an amino group. So together, they make up the basis of why we call these kinds of molecules amino acids. So if an amino acid is a molecule that has this group and this group, well, it seems as though you could make any kind of molecule with those 2 groups and it would be an amino acid. But when it comes to biochemistry for the purposes of the MCAT exam, amino acid refers to what we call alpha amino acids, which is where both the amino group and the carboxyl group are bound to the same carbon. So this, again, this angle is implying the carbon atom, and both this carboxyl group and this amino group are bound to that atom. That is called an alpha amino acid. Alpha amino acid is a molecule with amino to the same carbon. So when the amino and the carboxyl group are bound to the same atom, the same carbon atom, that carbon atom is called the alpha carbon. Okay, so before I get into the nature of that carbon and what makes that carbon so important as sort of the center of our molecule, I just want to show another depiction of this fundamental amino acid model. So here you have the amino group on the left. This is labeled. This is a figure from the textbook that I referenced earlier. So this is the amino nitrogen with 2 hydrogens. This is the alpha carbon, as they're pointing to here, which has 1 hydrogen and one what we call an R group or a side chain. And I'm going to mention that, but I just want to get to the other side here first. And then this is the carboxyl group, which is a carbon, an oxygen, and hydrogen. So first, the R side chain is so called because one can substitute that group for many different things. Specifically, for the purposes of the MCAT, there are 20 R groups that you need to memorize. And each of these R groups is part of a unique amino acid that has a unique name. And we're going to get into those names in section 8 when we talk about names and abbreviations of amino acids. But this is the model. There's 20 of these different R's, and every amino acid has a different R, and it's what defines them. So from now on, I'm going to be calling alpha amino acids simply amino acids because that's how they're referred to on the MCAT exam. But be aware that any molecule with a nitrogen like this, a carboxyl like this, could potentially be an amino acid. Okay. So first there's this R group that I just explained, and then there's seems to be a difference between these two drawings. This is the one that I drew, where this N has a plus next to it and 3 hydrogens, and this O has a minus next to it and no hydrogens. And this drawing, where the N has 2 hydrogens and no plus, and the O's don't have any negative, and there's an extra hydrogen connected to that O. So why don't I get to that in section number 3? But for now, keep in mind that both of these are alpha amino acids, and both of these are the definition of an amino acid because they are a carboxyl and an amino bound to the same carbon. Okay, so let's talk about that alpha carbon between those two functional groups, between that amino functional group and that carboxyl functional group. The alpha carbon is bound to 4 groups. And that is the case in every single amino acid. 4 groups. So this alpha carbon right here, again, the carbon is implied by the angle, and I'm circling with the cursor here. That atom is bound to an H, the R group, which could be 20 different things, the carboxyl group, and the amino group. So counting that up, that's 1, 2, 3, 4 different substituents. So in chemistry, its substituent is any group that is bound to the group that you are referring to. So these are the different substituents of any amino acid. There's 4 of them. So this is a concept, this brings to mind a concept in organic chemistry, where when you have a carbon atom that is bound to 4 different groups, that carbon atom exhibits something called chirality. So every carbon is going to have 4 bonds in its neutral, or let's say ideal state, 4 bonds to 4 different groups, But when those are all different from each other, So let's say this R group was um, a CH2CH3 chain, so carbon, 2 hydrogens, carbon, 3 hydrogens chain. That would mean that the carbon in the middle here, the alpha carbon, is bound to 4 different groups. And when you have a carbon atom bound to 4 different groups, it is what we call a chiral carbon. So the alpha carbon is a chiral carbon. So when it comes to a chiral carbon, or a carbon that is bound to 4 different groups, you are going to have 2 forms of that molecule that look sort of similar, but are different in the arrangement of the atoms that make up that molecule. So a chiral carbon means that you have at least 2 different forms of that molecule that differ in the 3-dimensional arrangement of their atoms. So in this picture, the hydrogen is at the end of this black, what we call a wedge, and the R is at the end of this, what we call a dash. So the wedge means that that hydrogen is popping out of the plane of the screen or the paper or the whiteboard that the molecule is drawn on, and it's coming towards your face. The dash means that that atom is going into the plane of the screen or the paper or the whiteboard, and it is going away from your face. So the molecule I drew here has 2 groups that are on the surface that you're looking at when you look at the molecule, and that is the amino group on the left and the carboxyl group on the right. Then it has the hydrogen, which is coming out towards you, and then it has the R group, which is going past and it's going away from you. So, such is the nature of the carbon atom. It's a 3-dimensional structure, and when you scale that up to an organism like a human being, you have a 3-dimensional structure. So that sort of thing is reflected all the way down to the molecular level for us. So given that this is 3-dimensional, you have 2 different forms of this molecule. They look similar, but they are different. And those are called, this is another depiction of the same thing. Those are called isomers or stereoisomers. And particularly when it comes to the 3-dimensional arrangement of 2 molecules that have all of the same atoms, but they just differ in the way that they're shaped 3-dimensionally, they are called enantiomers. And when it comes to categorizing enantiomers, there's the system of R or S. There's also the system of D or L. I'm going to explain both of these now and explain where amino acids fit in. So the R and S system is based on the arrangement of atoms in space. The D and L is based on how a crystallized, or no, dissolved solution of that molecule reflects or refracts rather plane polarized light. So in organic chemistry, we learned how when you shoot polarized light through a sample of this particular kind of molecule, that polarized light will sort of uh, bend at an angle, and that angle can either be positive or negative, and that determines whether it is D or L. So these two systems of categorizing chiral molecules are based on different properties of the molecule. And for the purposes of the MCAT exam, I recommend memorizing which ones they are for amino acids rather than figuring it out every time you see that molecule depicted somehow. Dates plain as light. Okay. So for amino acids, I'm going to double check with the miles down sheet that I mentioned before. For amino acids, each amino acid has an S configuration. So when it comes to R or S, it's going to be, I'm going to bold this, all S except cysteine. And then when it comes to D or L, it's going to be all L. I think okay. So that covers the alpha carbon. The alpha carbon of the amino acid, again, is the carbon that connects that carboxyl group and the amino group. And it has this really important property in that it is chiral, which means it is bound to 4 different chemical groups. That means that it has 2 different forms, at least, R or S, which can be categorized as R or S, and D or L. And we also are going to memorize that amino acids found in our body are all S, except for this one called cysteine. And they are also all L, including cysteine. Okay. Topic number 3 is the NH3 plus and COO minus groups. That brings me back to the pictures I was showing earlier. This is the one that I drew, and this is one from a textbook, which depicts the amino acid backbone, or the building, the scaffolding upon which amino acids are built. So in this picture, you see these 2 charges, a positive and a negative. And in this picture, you see no charges. That is because this is what you would call the ionized form of the amino acid, which means that it has ionic properties, i.e., it is charged. It is a charged molecule, or a molecule with charges. But knowing the most basic electrostatics, it's 1 positive charge here, 1 negative charge here. Overall, this is a neutral molecule. The same is true in this picture. This is a neutral molecule overall. Let's just forget about the R group for both of these pictures. This is a neutral molecule. So what's the difference between this and this? This one is ionized because there are charges, but they cancel out. And this one is not ionized, and so there are no charges. But let's stick up here for a second. And there is a term or a vocab word that we use when we're talking about a molecule that has charges, but whose charges cancel each other out, such as this one. And that is a Zwitter ion. So it's a German word, which I believe I read on Wikipedia means hermaphrodite. Okay. It means an ion that is not overall charged, where the charges cancel out, and particularly in a molecule, and in this case, an organic molecule that has equal and opposite positive and negative ions, or charges rather. So that is a Zwitter ion. And then you have this one, which is not ionized at all. Not ionized. Well, why do we have these two depictions? Is it one or is it the other? Really, it's this one, and that's why I drew this out as my definition of the amino acid. And that is because amino acids are ionized when they are in water, i.e., an aqueous environment. Such an environment, water-based, is found in the human body. So of these two pictures, which one will you see if you're in the human body? You're going to see this one. This doesn't really exist. So let's go a step deeper. Why are they ionized when they are in water? And particularly, why does the ion look like this? Well, let's see another difference between these two pictures. There's one H on this N that is not there here. And there's one H missing from these O's, which is there here. The transfer of those H's is the domain of acids and bases in chemistry. An acid loses an H and a base gains an H. So that, I think, brings us to our next, well, it sort of brings us to topic number 5, which is acid-base properties, but we're just briefly going to mention it in the context of talking about topic 3, the NH3 plus and the COO minus groups. So it turns out that this is an acidic group. That carboxyl group is acidic. So it gives its H, right? There's the H before it loses it, and there's the H after. It's not there anymore. And then a base takes an H. So there's the nitrogen without an extra H, and then there's the nitrogen with an extra H. So this is an acidic group. So when it is in water, it is not going to have its H anymore because it gave it off. And this is a basic group because when it is in water, it grabs an H from water, which is H2O, and it becomes this. So then why the charges? Well, an H is a hydrogen ion, which is a proton. And a proton is a positively charged particle. So when you gain an extra proton, or H, I'm going to use them interchangeably now, you gain a positive charge because protons are positively charged. When you lose a proton, or H, such as this group went through, you lose a positive charge. So with electrostatics, you lose a positive, now you're more negative. You gain a positive, now you're more positive. So that's all that happened here. You lost a hydrogen here, it's negative. You gained a hydrogen here, it's positive. And then you have your Zwitter ion, which is a molecule in which positive and negative groups are equally and oppositely charged, and the overall molecule is neutral. Okay. So I'm going to put these under here. So amino acids in the body are going to be in their, in their Zwitter ion form. Okay, and before getting into more about the acid and base properties, I want to move past this simplified drawing of an amino acid, where we just put R as a as basically a sit-in for any molecular component of the 20 that may be attached to this backbone, And I want to talk for a minute about those different molecular components and sort of what they look like. Okay. So this is a figure that I got from MIT OpenCourseWare. So it's a free course that they offer on YouTube that is part of the playlist I mentioned before. And I chose this because it gives a less user-friendly depiction of the amino acids, which in my experience is what you find on the MCAT. It's not a simple picture of the molecule that gives it in a sort of digestible way like this. It's usually a little bit more convoluted. And so this lesson will, in essence, be practice of seeing the amino acid in this different sort of context, but realizing that it's the same molecule that you memorized when you memorized the amino acids for the purpose of the test. So the R groups. This is that carboxyl group that is depicted further up in the document. This is the amino group. This is the alpha carbon. And this is the R group. So this, what I'm circling around right here is an R group, alanine. That group is a CH3 or carbon with 3 hydrogens. This amino acid has an R group, and this is called valine. So that is 1, 2, 3 carbons. This carbon has 1 hydrogen, that carbon has 3, that carbon has 3. Let's get a little more interesting. Okay, proline. So here is your carboxyl group. Here is your amino group. And this R group comes back and binds again to that amino group. So this is the alpha carbon, there's the R group, and that R group swings back and binds back to the nitrogen of the amino group. That is proline's R group. This is one of the basic amino acids, arginine. This amino acid has an R group that is a CH2, 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 and then NH, carbon double bound to NH2 positive. That's what makes it basic, just like the amino group on the backbone. And then an NH2 group at the end. So these R groups have different properties, as we've seen just by looking at a couple of examples already. And there's many ways to sort of divvy them up based on their properties. And we're going to do that now. So the first category that MIT puts these amino acids in, and I believe this would be, I think this is a better one for now. So let's go with this first category of R groups, amino acids with hydrophobic side chains. So the side chain and the R group refer to the same part of the molecule, which is that fourth thing that the alpha carbon is bound to. Okay. Hydrophobic side chains are side chains that are hydro-water phobic fearing, i.e., they do not want to bind with water and water-like molecules. Water being H2O, water-like molecules, including things like an alcohol, which has an OH group on it. These hydrophobic side chains are going to avoid being near those other kinds of molecules. So proteins can be very long chains of amino acids. And when a protein is done being formed by the ribosome and then other parts of the cell, the part of the protein that has those hydrophobic amino acids are going to be the parts of the protein that are in the middle of the protein. Whereas the ones that have hydrophilic amino acids, those are going to be the ones on the outside. And that is because the cell is a water like environment. These side chains don't like water, so they're going to avoid that environment. They're going to be on the inside. Whereas a side chain that is hydrophilic, which is essentially the rest of these side chains, is going to be more likely to be on the outside of a protein, and there are many exceptions. So these ones are the hydrocarbons, or just ones where there are carbons and hydrogens. This one has a sulfur atom. These have carbon hydrogen parts that are pretty big, but then they have other little groups, like the hydroxyl or OH here, or the amino or NH here. And then you have something that also is carbon and hydrogen, but it's special because, like I mentioned before, it binds back to itself. That's a cyclic or cycle or circle side chain. Okay, so now the rest of these are no longer hydrophobic. These are hydrophilic. So what makes them hydrophilic? It's their polar nature, or the fact that they have some kind of polar chemical bond as part of their R group. So this is the section about R groups. These are the ones that are hydrophobic, i.e. water fearing, and that is because they are not polar like water is. And these are the hydrophilic or water loving R groups because they are polar, just like water is. So I'm going to use one example is what I mentioned before, the fact that alcohol groups or OHs are like water. And so serine is a hydrophilic or water loving side chain. So I'm going to go back up to topic 4, the R groups, and I'm going to mention that that there are 20 different R groups. Before I move on from the R groups, I want to mention what the amino acid looks like when it is part of that polymer that I mentioned before, which is, of course, the protein. So amino acids are the building blocks of proteins, but what does it look like when they are actually bound to each other and not free, like I drew them before? So this is from the Miles Down study aid that I mentioned before. This is a series of amino acids that are bound together in a string. So the amino acid is the monomer, the string is the polymer, and each of these red R's represents a different R group. So starting from what we call the N terminus, I see the amino group that is part of any free amino acid. That amino group or N group is hanging off here because this is the end of the polymer. The polymer stops here on this side and it stops here on this side. So in essence, this whole thing is like one big amino acid where you have the amino group on one side and the carboxyl group on the other side. But inside that you have these bonds between the amino acids that I'm going to show now. And So take a look at the left here. The N terminus is attached to an alpha carbon, which is attached to a carbonyl carbon, which is the organic chemistry name for a carbon atom, which is double bound to an oxygen atom, and then another N for another amino acid, another alpha carbon, another R group, another carbonyl carbon, and then another N. So this R1 could be any of the 20 groups I showed up there. That R2 could be a different one, that R3 could be a different one, R4 could be a different one. They do follow certain patterns that is more the domain of a lesson on proteins and protein structure, but it could hypothetically be that you can combine any of those 20 amino acids together to make a very, very large number of different polymers. There are many possibilities, especially considering all of the different chemical changes that the body can make to a protein once it is done stringing along those amino acids. There's many chemical changes that the body can make to that molecule. But taking a look at these amino acids bound to each other, this carboxyl group where you have a carbon bound to two oxygens is no longer in the middle. I'm talking about now. So in the middle, you no longer have that carboxyl group where the carbon is bound to two oxygens. Now these carbonyl carbons, right? The carbon bound to the double O, yes, that's correct. Okay, are bound to nitrogens instead, and those nitrogens come from other amino acids. So this carbonyl carbon is to one nitrogen, and then another alpha carbon, carbonyl carbon, nitrogen, alpha carbon, carbonyl carbon, nitrogen, alpha carbon, and then the carboxyl terminus. This functional group, where there's a carbon with a double bond to oxygen and then a single bond to nitrogen, is called an amide group or a carboxylic amide. In the context of an amino acid, it is also called a peptide bond. So I'm going to write those both in the discussion of R groups. So just making sure the sound is still on. Okay. So the peptide backbone that is every part of the peptide or protein rather outside of the R group is bonds between different amino acids, aka peptide bonds. So peptide bonds form peptides, which is the name of specifically a smaller chain of amino acids or a chain of amino acids that is not very long, such as this one. This is a polypeptide. So that it refers to a relatively small protein. Proteins get very, very big, especially the most important ones like hemoglobin, for example. This doesn't even begin to sort of encapsulate the diversity. But this is the peptide bond, and that is the bond between amino acids when they come together to form a protein. That bond is an amide functional group, i.e., a carbon double bound to oxygen, single bound to nitrogen. That is an amide functional group. Okay. Returning back now to the diagram of all the different side chains. MIT has these depicted as their Zwitterion forms because that is how they exist in a water-like environment. But what kind of water-like environment is the human body? Well, when it comes to a discussion of a molecule that can either accept a hydrogen, a base, or donate a hydrogen, an acid, there's a term that can describe the different kinds of environments that those molecules create, which is the pH, or the measurement of how acidic that environment is. So H plus, which is also a proton and also a hydrogen atom, is measured in moles, which is just, let's say, like number of atoms per liter. It's number of atoms multiplied by a much larger number. So moles per liter. And that is a concentration. And then to further simplify things, we express the concentration of hydrogen atoms as a P of H, and P means the negative logarithm base 10 of the H plus concentration. So it turns out that the concentration of protons or hydrogen atoms is such a small number that it would be inconvenient to have to state that particular number every time we talk about how acidic or hydrogen, how concentrated with hydrogen a certain environment is. It turns out that those numbers would be really annoying to say. It would be something like 1 times 10 to the negative 7. Such a small number, moles per liter. Such a small number and really long to say because you need to express it in exponential form by saying 10 to the negative 7. Another way of saying it is 0.000007. That's why 7 moles per liter. Both of these are pretty weird and inconvenient, particularly because the acidity of the human body is a very important measure for professionals to discuss and to communicate about. So when there is a measure that is that important, we tend to a more simple expression. So the P, or negative log, of that concentration, that big unwieldy number, is going to be a much more simple number to say. So let's say negative log of 1 times 10 to the negative 7th power is going to equal 7. I'm going to quickly recap or describe what this function is, the logarithmic function. So for this lesson, the logarithm is an expression of an exponential number, and that expression is asking how many times do you have to multiply 10 by itself to get the number in parentheses here. And so then the answer is 7 times. So 1 times 10 to the 7 is 10 to the power of 7, or 10 times 10, 7 times. So it's a 1 with 7 zeros. When you go with negative, you move the decimal point in the opposite direction. So 1 times 10 to the negative 7 is negative 7. Or log base 10, rather, of 1 times 10 to the negative 7 is negative 7. And so to further simplify things, because we don't want to have to use negative numbers all the time, we say that negative log base 10 of 1 times 10 to the negative 7 is 7. So we just take the negative of both sides, and then it becomes this number. So 7 is the neutral pH. The body's pH is 7.35 to 7.45. Checking that, yeah. So on Google it says the same thing. Yeah, the body keeps pH in very tight regulation because, like it is depicted here, all of these molecules are going to be trading these H's, and since all of these molecules are the building blocks of your body's structure and function, when those H's are out of balance, both the structure and function of your body sort of begins to collapse. And so the body keeps the hydrogen, or those positive H's, the number of those, the concentration of those, in very tight regulation. And specifically, those are pH 3.5 to pH 7.35 to pH 7.45. That is what is called physiological pH. So this is part of the discussion of the acid-base chemistry of amino acids. And so when we have a pH of about 7.4, that amino group is positively charged. It's accepted an H, and that carboxyl group is negatively charged. It's given off an H. And that is because this environment has a concentration of hydrogens such that this group has acted as an acid and this group has acted as a base. But how do we quantify, like what number can we use to determine, given the hydrogen concentration of the environment expressed as a pH, how can we then determine what our molecule looks like in terms of the arrangement of its hydrogens, whether it's given one, accepted one, and which groups do what? Well, we have a number for that, and that is the P of the Ka. So an acid-base reaction whereby a hydrogen atom is either donated or accepted is like any other chemical reaction in that it can be expressed as an equilibrium between the molecule with and the molecule without the hydrogen. And so the products are going to be molecule without hydrogen, and the reactants are going to be molecule with hydrogen and water, because this is all happening sort of in water. A way to express the side of the reaction that your particular acid or base prefers based on its chemical properties is the Ka, or the rate constant of that reaction. And just like we do with the hydrogen concentration, rather than express those rate constants as those big unwieldy sciencey numbers, we use the negative logarithm of the Ka, or the rate constant of the dissociation of the acidic or basic chemical species into its constituent hydrogen and acid or base, and then water without hydrogen. We use the P, the negative log. So it's pKa. So let's ballpark the pKa of that carboxyl group as about 2, and the pKa of that amino group as about 10. Without getting into what exactly those numbers mean, that's sort of up here. When the pH of the environment is higher than a chemical group's pKa, that chemical group loses hydrogen. When the pH, or the chemical environment of that amino acid is, or that functional group or molecule is lower than the pKa, that molecule or functional group loses a hydrogen, or doesn't have a hydrogen. Plus pKa, COOH, pKa. So that for the NH3 plus, let's say it's 10, and for the COOH, let's say it's 2. Numbers might be given on the MCAT, or they could be memorized for Okay. Yeah, so that's about correct, based on my quick Googling, but again, those sources that I listed above are the more reputable sources. Definitely better to consult in this video. But let's say that the pKa of the COOH is 2, the NH3 plus is 10, in an environment in which the pH is 7, or the concentration of hydronium ions is 1 times 10 to the negative 7 moles per liter, you will have a negative COO here, a positive NH3 here, and then you have your R group. Well, it turns out that some of those R groups, just like the amino or the carboxyl of the backbone, those R groups can also be acids or bases. So when it comes to something like aspartic acid, there is an acidic R group that, just like the backbone, has given off hydrogen and become negative. And then with an amino acid like arginine or lysine or histidine at a pH of 7, the R group has a basic functional group that has gained a hydrogen. So for that, that's this NH3. And for that, that is that NH2, where the N, since it's double bound to that carbon, is positively charged due to its being bound to 4 different atoms. Okay, so that is the acid-base properties of the amino acid. No acid backbone has an acid and a base. So the acid is COOH, and the base is NH2. No acid R groups. So acidic would include something like aspartic acid. So the acid part is in the name. And the basics can include things like lysine, or amino acids like lysine, rather. So rather than just being relegated to or acid-based properties, rather, amino acids also have special properties. Now, these go far deeper than the medical college admissions test is going to test, but there are a few that are commonly tested and also foundational. The first one I want to mention is the R group on this amino acid, which is cysteine. So I'm going to actually screenshot that and paste that as the first subpoint in my topic number 6, special properties. So that is going to be the disulfide bond between cysteine amino acids. So cysteine is the amino acid with the R group CH2 and then SH. Those SH groups on cysteine can bind with each other. So one cysteine and another cysteine bind to each other, and they form together what is called a disulfide bridge. And when two cysteines combine their sulfurs to form the disulfide bridge, they are now named cysteine. So a way I liked to memorize this when I was studying for the MCAT is that the longer name, or cysteine, is for the smaller thing, the thing where the two amino acids are not bound, and then the bigger thing is the smaller name. So the bigger thing where they are bound together is the smaller name, which is cysteine. So it's missing an E, a very subtle difference, but the sort of thing that test takers could exploit since they often like to exploit sort of differences in language, very subtle differences like that. Okay, so the first one I want to mention is the disulfide bridge. The second one is hydrogen bonding. And I would put this actually under a category of the properties of the OH functional group. So number one is going to be hydrogen bonding. The human body is a water-based environment. A property of water that defines many of the ways in which it acts as a chemical is hydrogen bonding, or the bonding between the oxygen of one OH group and the hydrogen of another OH group. This can actually happen with an OH, NH, or FH group. So I'm going to use this amino acid as my example for hydrogen bonding, because in its R group is a CH3, which is pointing out from us in this picture, a CH3, and an OH. And since that OH has an oxygen bound to a hydrogen, it is going to be capable of hydrogen bonding, which is an interaction that water molecules perform between each other, and that a threonine molecule, due to its R group, can perform with another threonine molecule or any other molecule with an OH, FH, or NH functional group, hydrogen bonding. Another property of the OH functional group is phosphorylation. So I'm going to paste this image that I just searched up on Google Images of the phosphorylation of two OH containing amino acids. This first one is serine, which has the R group of just OH, and the second one is tyrosine, which has the R group of an 6-carbon conjugated ring, aka an aromatic ring, and then its OH group. Well, focusing in on the OH, in both of these cases, phosphorylation turns an OH into an OPOOO. That reaction where the OH is turned into an OPOOO is important in countless biochemical processes that occur within the human body. And so it is important to note that when we're talking about threonine, serine, or tyrosine, amino acids that have OH groups as part of their R group, the possibility of phosphorylation is there. I want to quickly check. I want to just check to see if there are any additional properties that I might have missed. Okay. One I want to mention about proline is its shape. Is acid whose R group binds to the backbone. Proline is going to serve a distinctive functional structural function in proteins because of the way that its R group twists around and binds back to the backbone. That is proline. That is important to mention, and more important when it comes to talking about the structure of proteins, but also relevant in the discussion of amino acids, in my opinion. Before moving on and talking about memory tricks or just memorizing the names and abbreviations of all the amino acids, I want to briefly touch on the alpha carbon. So a special amino acid is the first amino acid that we're going to be memorizing, and so we might as well memorize it right up here, and that is glycine, which is the amino acid, the R group of hydrogen. So in topic number 2, which was the alpha carbon, I mentioned the chiral nature of the alpha carbon. A chiral carbon being a carbon atom that is bound to 4 chemical groups, which are all different. 4 different chemical groups makes a chiral carbon. But what if that R group that is here on the dash or facing away from us in this depiction that I drew for this lesson, what if this R group is just a hydrogen atom? Such is the case with glycine. So I don't know why MIT decided not to put glycine there, but whatever. Okay, here, this is another chart of the amino acids. It is very helpful to see different charts of the amino acids because the MCAT can depict amino acids in different ways depending on how it wants to test you. I find that they often want to test your recognition of something in an unfamiliar format. So this is glycine, depicted a little bit differently from the MIT diagram above. You can see that the R group is in hydrogen. So is this carbon bound to 4 different chemical groups? No. 2 of them are the same, 2 of them are hydrogens. That means that it is not a chiral carbon. So the very first amino acid that we're going to memorize, which is glycine, is the only amino acid that displays none of this RNS stuff. So not a chiral carbon. And so I'm going to also put that in the properties, the special properties of amino acids, glycine. Glycine is not chiral. Okay. Okay, now the names and abbreviations of the amino acids. For this part, I'm going to go back to the miles down review sheet, and I'm going to go to, I'm in another tab now. Now that I'm in the miles down review sheet, specific the part about amino acids, I'm going to go through and use this as our tool for memorizing these structures because I find that this is the most straightforward diagram that I've seen about the amino acids. That is because all of the atoms here are actually written out. Rather than there being an angle here that implies a carbon, there is a C, and it is connected with these lines to 2 different H's, and that represents glycine. So this is going to be the chart that we use to do the memorizing. And I like to start with the non-polar, aka hydrophilic, or hydrophobic rather, water-fearing amino acid side chains. So this is the first of the 20 amino acids. One is glycine. Glycine has the 3-letter abbreviation. So before I go into that, I want to say that the amino acid names are abbreviated in 2 different formats. One is a 3-letter abbreviation, and one of them is a 1-letter abbreviation. The MCAT tests knowledge of the 1-letter abbreviation, the 3-letter abbreviation, and the name. So those 3 signifiers need to be memorized for every single chemical structure here. So each of these 20 chemical structures has a different set of those 3 things: the full name, the 3-letter, and the 1-letter. Number 1 is glycine. Glycine is the simplest because our group is a hydrogen atom. The second most simple amino acid is an alanine, which has the R group of a carbon bound to 3 hydrogens. Another name for a carbon bound to 3 hydrogens is a methyl group. So this is alanine, and it has a methyl group. Then there is valine, which has C bound to H and then bound to 2 other C's. So it sort of forms like a V. So that's valine. We're still hydrophobic here. This is leucine, which is like valine, except it has a CH2 between, or a CH rather. Sorry, it has a CH2 between the carbon that's bound to those 2 carbons and the alpha carbon, which is sitting up here between the amino group and the carboxyl group where it belongs. After leucine comes isoleucine. Isoleucine has the same atoms as leucine, except that one of those 2 CH3 groups is bound to the carbon right after the alpha carbon rather than to the carbon at the end of the chain here, like it is with leucine. Isoleucine suggests that it might be an isomer, or a same number of atoms molecule but different configuration from leucine. So, and that is that's the case. It is an isomer of leucine, isoleucine. Next, I would memorize phenylalanine. Phenylalanine has a CH2 and then a 1, 2, 3, 4, 5, 6 carbon ring that is aromatic. So a few names for this same thing here is an aromatic ring, a benzene ring, and a conjugated 6 carbon ring. All of those names that I just said describe the same thing, and that is indeed what is at the end of the R group on phenylalanine. Next is cysteine, which has a CH2 and then an SH. Another term for that SH group is a thiol, THIOL group. So just like we have alcohol for OH, we have thiol for SH. Then proline, which has that R group that bends back from the alpha carbon and attaches to the amino group of the amino acid backbone. So phenylalanine, cysteine, proline. Then methionine, which has 2 CH2s, a sulfur just like cysteine, and then after that sulfur has a methyl group or a CH3 group, just like alanine has a methyl group, CH3. That's methionine. So I'd like to think of it as like methyl, thio is the sulfur, that's a thiol group, and that's a sulfur, so methionine. And then tryptophan. Tryptophan has a 2-ring aromatic conjugated structure in its R group. This one is a little bit more difficult to describe in words and is better seen in a diagram like this when it comes to memorizing. For glycine, the 3-letter abbreviation is GLY. The 1-letter abbreviation is G. The R group is a hydrogen. For alanine, the 3-letter abbreviation is ALA. The 1-letter abbreviation is A. The R group is a CH3. For valine, the 3-letter is VAL. For valine, the 1-letter is V. The R group is CH, CH3, CH3. For cysteine, the 3-letter abbreviation is CYS. The 1-letter abbreviation is C. The R group is CH2SH. For proline, the 3-letter is PRO, and the 1-letter is P. And the R group is CH2, 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 3 members of a 5-membered ring, which includes the alpha carbon and the NH2 group itself. or the amino group of the backbone of the amino acid. Leucine's 3-letter abbreviation is LEU. The 1-letter abbreviation is L. Leucine's R group has CH2, CH, CH3, CH3. Isoleucine has a 3-letter abbreviation of ILE and a 1-letter abbreviation of I. Its R group is CH, bound to both a CH3 and a CH2, and then that CH2 is also bound to a CH3. Methionine's 3-letter abbreviation is MET, and then its 1-letter abbreviation is M. Its R group is a CH2, CH2, S, CH3, all in one chain. Tryptophan has a 3-letter abbreviation of TRP, a 1-letter abbreviation of W, and a R group that includes a CH2 and then a 2-ring structure, one of which is a 5-membered conjugated carbon ring, which includes one amino group as one of the 5 members, and then a 6-membered conjugated carbon ring with only carbon atoms. Speaking of 6-membered conjugated carbon rings, phenylalanine has a 3-letter abbreviation of PHE, a 1-letter abbreviation of F, and an R group that has CH2, and then a benzene aromatic 6-membered conjugated ring. After the nonpolar or hydrophobic amino acids, on this diagram is the category of the positively charged or basic amino acids. Basic because they accept hydrogen atoms, aka protons, and those protons being positively charged give the atom an overall positive charge, thus the category in this diagram of positively charged amino acids. Lysine has this R group with 1, 2, 3, 4 carbons, all of them with their correct amount of hydrogens, which would be 2 for each because they each have 2 bonds to other carbons. And then at the end of those 4 carbons comes an NH3 plus group, much like the one that is found on the amino acid backbone in the amino group. So that is lysine's R group. Then there is arginine, which has a CH2, 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 3 carbons, then a nitrogen with a hydrogen bound to a carbon. That carbon is double bound to that nitrogen that I just mentioned, another nitrogen, to which it has 2 bonds, and then a third nitrogen, which it has 1 bond to. So it's carbon with a bond to the NH that leads back to the alpha carbon, another N, which is double bound to that carbon, and then bound to 2 hydrogens, which makes it positively charged because it has 1 extra positive charge. And then it's also single bound to an NH2 group, which is neutral at the end of the molecule. That's arginine. Histidine has the R group of a 5-membered ring that contains carbons and 2 nitrogens. So if it's 5-membered, that would mean there's 3 carbons and 2 nitrogens, and it is a conjugated ring. So there's double bonds that link the carbon, nitrogen together, and it has the appropriate number of hydrogens for that situation to be the case. Lysine has the 3-letter abbreviation LYS and the 1-letter abbreviation of K. Arginine has the 3-letter abbreviation ARG, ARG, and the 1-letter abbreviation of R. Histidine has the 3-letter abbreviation HIS and the 1-letter abbreviation H. Moving on to the opposite of nonpolar, polar, and the opposite of hydrophobic, hydrophilic. These are the polar hydrophilic amino acids. First, there is serine, which has the R group CH2 and then OH. That OH, as mentioned before, participates in hydrogen bonding and is also a possible site of phosphorylation, or the conversion of an OH group to a phosphate group, which is important for many biological processes. After serine comes threonine, which is like serine in its R group, except that C that comes off of the alpha carbon is bound to a hydroxyl, an OH group, and a CH3 group. Then there's tyrosine, which between the alpha carbon and the OH group has a CH2 and then an aromatic six-membered carbon ring. Tyrosine. After tyrosine is asparagine, which has a CH2 and then an amide group, much like the peptide backbone of a protein when amino acids bond to each other, an amide group. An amide functional group is characterized by a carbon double bound to oxygen and single bound to an amine group, or NH2. After asparagine is glutamine. Glutamine has that same amide group at the end of its side chain, except between that amide group and the alpha carbon, unlike with asparagine where there's only one CH2, there's two CH2s. So glutamine has two CH2s, asparagine has one CH2 in its R group. After, so let me do the abbreviations. Serine has the three-letter abbreviation SER and the one-letter abbreviation S. Threonine has the three-letter abbreviation THR and the one-letter abbreviation T. Tyrosine has the three-letter abbreviation TYR and the one-letter abbreviation Y. Asparagine, that shorter amide one, has the three-letter abbreviation ASN and the one-letter abbreviation N. Glutamine has the three-letter GLN and the one-letter Q. Rounding that out, glutamine is the amino acid with the amide group and two CH2 groups between that amide group and the alpha carbon. The final category of amino acids, as shown in this diagram, is the negatively charged amino acids. These amino acids are such because they have R groups that have lost a positive charge in the form of losing a proton, or an H+. Losing a proton is what defines an acid. So these are negatively charged. They are acidic as well. So their negative charge and their acidity is describing the same property of that structure to lose a hydrogen atom. So aspartic acid has the side chain CH2, C double bound to O, single bound to O, just like the backbone, that is a carboxyl group. Glutamic acid has the same side chain, just like the relationship between asparagine and glutamine, except it also has another CH2. So it's just like asparagine and glutamine, it also has a CH2 between the alpha carbon and the functional group at the end of the side chain, which is now the carboxyl group. So aspartic acid is the smaller, glutamic acid is the larger. Another way of naming these last two amino acids is aspartate for the anion form of the molecule, which is actually the one that's depicted here, even though they don't call it that. So there are some issues with the miles down sheets. This, I believe, is one of them. And then glutamate, which is the name for the anionic form of glutamic acid, which again is depicted here and not named as such. But so this is aspartic acid, aka aspartate. This is glutamic acid, aka glutamate. Aspartic acid has the three-letter abbreviation ASP and the one-letter abbreviation D. Glutamic acid has the three-letter abbreviation GLU and the one-letter abbreviation E. After the live stream, I'm going to put those three letters, one letter, and full names into the document that is the sort of the home base of this live stream. So back to that home base, category 8 is the name and abbreviation of amino acids. And here I put the instructions. Memorize these. The name, the 3-letter, and the 1-letter abbreviation. And also all of the R groups. These come up on every MCAT exam, including the one that I'm going to show in the last part. Before I move off of topic 8, though, I want to mention how I memorized these amino acids. I started by doing what I just did in this video, which was going through the structures and learning what makes each amino acid unique. It's the R group, but specifically what different kinds of R groups are there? One could imagine any manner of organic molecule attached to that amino acid backbone. Which are the 20 that nature has chosen as the side chains of the building blocks of proteins that our genetic code encodes for? So it's those 20, and that's what I would recommend first, understanding what amino acids exist. After learning the amino acids, which we just went through and did, this comes to the domain of mnemonics. So figuring out some kind of real world, or better yet, a personal sort of naming trick to memorize those abbreviations and those names. So I'm going to go right for the most difficult one, which I think is tryptophan. Personally, that's my personal favorite, most difficult one. And I'm just going to go through how I memorized the name of the 3-letter and the 1-letter for that one. I'm going to go find it in this document, tryptophan. Tryptophan has a very hard time hiding because it's sort of like big and scary, and that's why it's my favorite one. So, all right, Google Docs. Okay. There is tryptophan. So I didn't come up with this mnemonic, but tryptophan has the 3-letter abbreviation TRP. And there's no mnemonic for that one. That's straightforward. But the 1-letter abbreviation for tryptophan is W, and that can be memorized by memorizing tryptophan. Okay, the amino acid tryptophan has a W as its abbreviation. There you go. There's 19 more, and they can be easily split up over days, weeks to be memorized. And as long as they are all readily accessible in the brain by test day, the test taker should be good. Okay, AAMC questions. Now I'm going to show official questions from the Association of American Medical Colleges, which writes the MCAT exam, specific questions that they have published that are about amino acids. So I'm going to start with this one. This is copied from the What is on the MCAT PDF. And I'm going to zoom in on the question and the answer choices so that the viewer of this video, I just want to make sure about this, the viewer of this video can't readily see the answer right now. The question asks, what type of functional group is formed when aspartic acid reacts with another amino acid to form a peptide bond? What type of functional group? A functional group is an arrangement of atoms that is seen across many different organic molecules. An example of a functional group is answer choice A, an amine group, which would be a nitrogen bound to a carbon, which is the organic molecule, and then bound to two hydrogen atoms and with a lone pair of electrons. That's one functional group. So what type of functional group or that category of things is formed, is created or results from a reaction whereby aspartic acid, aka aspartate, which is the anionic form of that amino acid, and that amino acid has the R group of CH2 and then the carboxyl group. So CH2, C double bound to O, and then single bound to an OH, reacts with, so that is an example of having it at your fingertips. That needs to be done by test day. With another amino acid to form a peptide bond. So what type of functional group occurs when aspartic acid and another amino acid form a peptide bond? This could go for any bond between amino acids. As mentioned before, when one amino acid binds to another amino acid, it forms an amide group or a carboxylic amide. So that is indeed the answer. And the AMC's explanation of this is pretty patronizing. I wouldn't read the AMC's explanation for all the questions. I mean, skim through it. But anyway, the functional group that's formed when one amino acid binds to another is an amide group. And we know that because the definition of an amide group is the kind of group that you see when two amino acids come together. It's by definition a carbon double bound to an oxygen, single bound to a nitrogen, and that's what we see right here in a polypeptide or a multiple amino acid chain. Okay. The more interesting AAMC question is one that here's a more complete AAMC example. I'm going to read through the passage quickly, and then I'm going to answer the question with the help of the passage information. So taking a giant leap now from content review to practice problems, which is the general format of any MCAT preparation plan, going from the content, from the theory to the practice, the question answering. Here's an MCAT passage. This is taken from a real medical college admissions test exam. Somebody who is now a doctor answered this question. Okay. Aluminum phosphide, AIP, is a pesticide that can react to produce phosphine, a gas that is highly toxic to humans and animals. Reaction 1. I see a reaction diagram where we have ALP, aluminum phosphide, plus 3H plus, or protons, goes to aluminum, the cation with the 3 plus charge, plus pH3, which I guess is our phosphine toxic gas. It is toxic to both humans and animals. Okay. Phosphine toxicity is the result of interference with cellular metabolism. It is thought that phosphine reacts with important sulfur hydro groups in complexes 1. In complexes 1, NADH dehydrogenase, so that's another name for complex 1. Complex 2, succinate dehydrogenase. Complex 4, cytochrome oxidase of the electron transport chain, etc. Researchers conducted a kinetic analysis of the effect of AIP on cytochrome oxidase, which is one of those things that they just mentioned. So they conducted this kinetic analysis, aka an analysis of the speed of the reactions, of the effect of AIP, which is aluminum phosphide, which is also represented here, on cytochrome oxidase. Okay, this is a Lineweaver-Burke plot, so I'm going to read it. Figure 1, a Lineweaver-Burke plot of cytochrome oxidase kinetics in the presence and absence of AIP with cytochrome C as the substrate. A Lineweaver-Burke plot is a representation of the activity of one of these classes of molecules, which is enzymes. And particularly when it comes to the activity of enzyme, what's important to represent is the speed at which the enzyme catalyzed reaction is occurring. So these lines are representing the speed of the enzyme catalyzed reaction without A1P or without AIP or with AIP. So here we can see without it, looks like this, and with it, it looks like this. Researchers measured the level of ATP, the rate of ATP synthesis, and the rate of ATP hydrolysis in rat liver mitochondria. It is found that AIP exposure resulted in a 65% decrease in ATP levels and a 48% decrease in the rate of ATP synthesis. In addition, the effect of AIP on the activity of 3 ETC complexes was determined. That's represented in Table 1. The activities of these complexes were determined independently of each other. Okay. Table 1 says the electron transport chain complex activity in response to AIP. On the left here, it shows the ETC component, which is complex 1, complex 2, and complex 4, which were called out earlier here. And then control activity, some numbers, and then activity after A1P exposure. I'm a little confused as to whether it's A1P or AIP. It seems to like be, I don't know. Activity after A1P exposure in, I think that's supposed to be like units per milliliter. Okay, so 13. Okay. All right, so that's Table 1. I think an important thing to mention in the context of reading this passage is that the electron transport chain, aka the ETC, is a series of enzyme catalyzed reactions that produces ATP. So the goal of ETC is ATP synthesis, increase ATP synthesis, increase ETC activity, decrease in ATP synthesis, which occurred when this process was exposed to AIP equals decreased is due to decreased ETC activity. So ETC activity and ATP are essentially like the same thing in this passage. Okay. Based on the passage, which amino acid will most likely react with phosphine? Okay. So let's go back into the passage and see where we might find the answer to this question. Complexes, actually complexes. Phosphine. Okay, so phosphine is a gas. It's a phosphorus atom connected to 3 hydrogen atoms. Phosphine reacts with important sulfur hydro. Oh, there we go. Okay. So the question was not about what this experiment was actually testing. It was about you finding this needle in the haystack where they mentioned that the phosphine, which is this gas, reacts with sulfur hydryl groups in these things, which are proteins, and specifically within the category of proteins are enzymes. So, as mentioned at the top of the lesson, the amino acid is the fundamental building block of the protein. Proteins serve functional and structural purposes in our body. This is the functional purpose, which is to produce ATP, which is the energy currency of the cell. So the sulfur hydryl group within these proteins is going to have to come from somewhere, and since it's a protein, that somewhere is amino acid. So which amino acid has a sulfur hydryl group? That is cysteine. So let's find cysteine, and that is indeed the answer. CYSB. Okay. So they probably explain it. Let's read the AAMC's explanation for this one because it's probably very straightforward. The passage notes that phosphine reacts with sulfur hydryl groups. Cysteine contains a sulfur hydryl group on its side chain, allowing it to react. Let's go see a picture of that. The sulfur hydryl group on the side chain of cysteine. That is right there. So sulfur or sulfur, hydrogen, hydryl. Sulfur hydryl group. All right, that finishes today's lesson on the amino acids. There are a few important protein structure and acid base concepts that weren't covered in today's lesson. Another important thing that was not covered in today's lesson was the way that amino acids are chosen by the cell to succeed each other in forming a protein. So a few concepts that sort of surround amino acids, such as what happens before they're chosen for proteins, and then what happens once they're part of proteins, whether that's the acid-base chemistry or the shape of proteins themselves, was not covered in today's lesson. I'll also put in the description of today's video a couple of other maybe big topics sort of surrounding amino acids that we didn't get to today. But thank you for watching, and visit my website if you're interested in a free tutoring consultation with me.